Hello, my name is Victoria Lotabosin,
And my name is Camille Hayden.
and we are mentoring under Dr. Joseph D. Silva, Department of Biology of Norfolk State University.
Our topic of study was the cultivation of the Rotifer Philodina species using moss and water in the lab. What are rotifers? Rotifers are tiny microscopic organisms that have habitat in many fresh, brackish, and marine water sources, as well as in lichen, moist soil, and moss. Their most identifiable feature is their corona, a ciliated structure on their head which they use to capture food. It's not COVID-19.
So, the role of rotifers mainly lies in ecosystems, with them acting as a filtration and nutrient system, as base food in many food chains, and as a toxicity assessment measurement for aquatic water. If our study is successful, it means an easier and cheaper way to acquire food for aquaculture farming, as well as making it easier to study rotifers.
The purpose of this experiment was to produce a laboratory environment that will allow the growth and sustainment of rotifer populations over a long time period.
This study is part of an ongoing, 8-year long ongoing study that was originally started in 2012. We were able to collect data that has led us closer to the ideal rotifer environment.
This research was conducted virtually, with no students or professors meeting each other at any given point in time. The research was conducted by Dr. Joseph De Silva and was passed on to the STAR students, who also did their own research.
For methods and materials, the moss was collected from Chesapeake Bay, while the rotifers were collected from a bird bath in Suffolk. The rotifers were classified as class Philodina, Philodina roseola species. The rotifers were separated in counts of 10, 30, 50, and underwent 5 different treatment groups.
Rotifers were collected using a small mesh fish net lined with medical gauze, which was used to filter out debris. They were maintained in conical test tubes and counted using a P10 micropipette. We also used a mortar, which was replaced by a petri dish with a scored bottom that was scored by a glass cutting knife, and the population was checked every 7 days.
We did preliminary experiments involving distilled and distilled water and rainwater to act as controls to analyze what effect the moss and moss suspension did have on the rotifers. For the distilled water experiments, we had 2 of the 3 experiments showing results, with only 1 experiment actually showing positive rotifer growth above the original rotifer population count.
This is the rainwater experiment, in which only one of the 6 trials experienced an increase in population. Overall, the increase was not a significant change, and the rainwater experiments underperformed in comparison with the distilled water.
In the moss and distilled water experiments, many of the experiments and trials experienced poor results, with many resulting in 0 or very just above 0 or for population counts. It's important to note that not all experiments are listed in the table displayed to the right, as only the most significant and non-repeating were chosen to be displayed.
These are the moss and distilled water experiments, in which the mortar experiments were done to check the preference of tough bottoms and smooth bottoms, as the rotifers in past research has shown that they preferred rough bottoms over smooth bottom containers.
In the moss and rainwater experiments, they were one of the best treatment groups with very good results, with 2 of the experiments yielding rotifer populations that stayed near or actually exceeded the initial rotifer population counts. It showed that this was a potential rotifer environment.
The data shown here is the moss suspension and distilled water experiment. It was expected that there would be a drastic majority decline in the population, but that was not the case, as these experiments had high ending rotifer numbers that would succeed or approach their initial rotifer counts.
For the moss suspension and rainwater experiments, it produced the best result out of all the treatment groups, as it had the most consistent rotifer counts, as well as showing positive rotifer growth, with some trials exceeding their initial rotifer population count.
The main treatment groups also had relatively high rotifer population counts with the moss and distilled water experiments, while the best performing experiments were the moss suspension and rainwater, closely followed by moss suspension and distilled water, and then moss and rainwater. Despite moss suspension and distilled water having the higher count, its trials deviated from each other far more than its rainwater counterpart, which held more stable values.
For the issues to overcome, the rotifers are moving too fast, and common methods were proven to be ineffective, and so freezing them was the most efficient way to actually view them. Zero dilution had to be employed when we were collecting the rotifers to be used in experiments, as not doing so often resulted in too many rotifers to be counted efficiently. And the most dangerous issue we had was the contamination of the moss collected from outside, and so it was pre-soaked in distilled water and then observed for any rotifers that would have been spotted.
It was concluded that rainwater was more efficient than distilled water as it produced more stable results, and that rotifer count did not matter, as a 50 rotifer count was just as likely to yield the same results as a 10 rotifer count. Now, although some experiments displayed a positive rotifer population growth, the treatment group showed overall decrease in population. This leaves us with the conclusion that moss itself is not that reliable of a medium.
For future work, we plan to have a more consistent number of trials and a more efficient method of slowing down the rotifers, as well as having less variables in our treatment groups to better analyze the data. We also want to use yeast as a food medium to make the results of our moss more stable and make sure they were not starved out.
We want to thank our mentor, program director, and the NSF Foundations for funding the program.
These are the references we used for our research.
Are there any questions?